Meine sehr verehrten
Ladies and gentlemen, I wish you a fantastic evening. It's great to see so many of you here, and although the weather draußen is brilliant, it was great that you decided to come here into this awesome lecture hall to learn a little more about light in biology. That's the title of the 8th symposium of the Hector Fellow Academy, to which I welcome you most cordially. We have prepared a great evening. I have talked to some of the presenters already, and I myself would have liked to be in the audience. So I envy you for being able to sit now and listen with great attention. We will dive into exciting processes associated with photosynthesis. We will look at that invention a bit more closely. We will then have a look at how light in biology interacts with animals. Animals have their own ways of dealing with light. And we will learn something about how to look into the brain using light. Isn't that great? Well, basically, we have this great opportunity tonight to listen to such fascinating presentations because science makes it possible. Science helps us to understand the world a little better, to understand what's going on around us. Promoting science is one of the objectives of the Hector Fellow Academy, an interdisciplinary community of cutting-edge scientists, some of whom are here tonight. It's not just promoting science, it's also bringing all these different minds together and promoting the spirit of interdisciplinarity. I'm grateful that I managed to pronounce that word, to enable them to achieve the next breakthrough. In many areas, scientists are so highly specialized that people, when they talk to one another, find it difficult to understand because they are so highly specialized. So looking across boundaries, looking beyond the box, looking outside of one's own confines, it's a recipe for success, is a recipe for an interesting evening. Well, together is a very important word tonight. We will not just listen together to interesting presentations. You're also very warmly invited to participate in the discussion. We have a round of discussion at the end, and you can ask your questions. I will tell you exactly how the microphones work when you want to participate. So when you press the button, there is the red light on, and everyone can hear you. So be careful what you say, and you should probably start doing that only in the discussion round. And those who are with us in the live stream, there is a button on the right-hand side, and you can send us your questions through the chat. I'm certain that we will get a lot of feedback from those of you who watch us online. We will start with an introduction given by Professor Hegemann. It's a very complex issue, and we need a little introduction and a few guidelines. We have a great opportunity of listening to Professor Hegemann now, and he will build us a bridge to understand what is going to happen after that. But before that, I would like to welcome Professor Marc Erhardt, Director at the Institute of Biology of Humboldt-Universität, who will welcome you here and open the proceedings. Thank you and enjoy the evening.
Thank you very much, Dr. Häusser. Mr. Hector, Mr. Bleich, Mr. Schauer, dear colleague Professor Hegemann, dear colleague Professor Zouni, dear colleague Monyer, Dr. Sumner-Rooney, Dr. Elsner, Dr. Häusser. Distinguished members of the Hector Fellow Academy, distinguished guests from culture, politics and science, dear researchers, to everyone who's interested in science, dear students, ladies and gentlemen. It is a great pleasure and an honor to be here today as the Managing Director of the Department for Biology of Humboldt-Universität, and to be here to welcome you to today's symposium under the title Light and Biology. The Humboldt-Universität in Berlin strives to make science accessible to a wider audience and to promote the dialogue between science and society. It shall not be isolated from society, but we rather like to promote a public debate with our citizens. And that is exactly what the symposias of the Hector Fellow Academy strive to achieve. Through this dialogue, they strive to find solutions for current challenges. And that is why I'm so pleased to be here with you tonight. Today's symposium will address an issue that is so important for our everyday life: light. Light is the basis for our life on this planet. 3.5 billion years ago, microorganisms, cyanobacteria, produced oxygen, gaseous oxygen from carbon dioxide. And plants also use light for photosynthesis, producing oxygen. Animals, human beings are also responding to light in terms of their biological rhythms or behavioral patterns. And in order to underline the importance of light, the Department of Biology has set itself the ambitious target of establishing this new area, optobiology. And therefore, this new building for optobiology is so important, as it will open up new opportunities for us in order to research into light and different processes stimulated by light. This development represents how we strive for excellence, interdisciplinary research, which the Hector Fellow Academy and Humboldt University have in common. Tonight, we are here to take a deep dive into the fascination for light. And with us, we have some of the most renowned experts. That is why I am so pleased to welcome Professor Hegemann here tonight. He's the scientific host or organizer of tonight's symposium. He's an internationally renowned expert for photobiology and co-founder of Optogenetics, and one of the reasons why Berlin has become an internationally renowned center for optogenetics. Professor Athina Zouni is also here with us, a leading expert when it comes to photosynthesis at Humboldt University. Dr. Sumner-Rooney from the National History Museum, as well as Professor Monyer are also here with us tonight. I would like to thank everyone who made this event possible: organizers, sponsors, speakers, and of course you, dear participants, for being with us tonight. Without your interest in science, a symposium as this tonight would not have been possible. I would now like to wish you an inspiring, fascinating evening full of new insights and stimuli about new innovations when it comes to light and biology and other aspects of life. I now look forward to the upcoming fascinating presentation, and I hope you enjoy this evening. Thank you.
Dear Marc, thank you for the friendly introduction. And thank you, Mr. Häusser. Also, thank you to you. I would like to welcome Mr. Hector. Welcome to Berlin. And welcome to the board members of the Hector Foundation. I would like to say hello to all those of you who are interested in the issue of light. Welcome to the Friends of Humboldt University and the museum. And welcome, ladies and gentlemen. Marc Erhardt already mentioned the most important things we want to do tonight, but let me add a few more aspects. As long as people exist, sun has always been a kind of mystery, a symbol of the divine character of the world. She had always been there. It was always clear that we depend on the sun, and this has basically not changed. We know how the galaxies develop, but there are still mysteries. What happened before the Big Bang is something we don't know, and have no perception at all as to how it could have happened. It is just zero, nothing, nada. What we do know is that the sun is important for us. We depend on it completely. Gravitation is important because otherwise our planet would disappear into the icy spaces of the cosmos. We know that the heat of the sun is absolutely necessary. This is something we have been acutely aware for the past 6 or 7 years. The moment things start to oscillate more than usual, everything is at risk. 4 million years ago or so, nature has developed systems to turn solar energy into biological energy, a process we call photosynthesis. It was first the retinal dependent photosynthesis, and then the one depending on chlorophyll. And all this happened irrespective of one another with different types of bacteria. And then nature managed to combine the two types of photosynthesis, forming cyanobacteria, and it managed to remove electrons from water, to transfer molecules that are redox equivalents and are part of our circulatory system, so that protons then can be used to control processes within our bodies. The key process that has been not understood for the longest time is the one of how water is split into the different molecules, and why there are atoms developing, but also toxic material. And we will learn a bit more about it from my colleague Zouni. Sensorics is something similarly old on different levels. They control biological processes. Photosynthesis doesn't do that. You know the phytochrome that is responsible for plants when they grow leaves. You know the visual process that we use to orientate ourselves. So these are two processes, sensorics to grow and sensorics to orientate. And the reprocess is probably the most complicated one of all the sensoric processes in our own body. Charles Darwin several times reworked the chapter on eyes because his wife said this can't be true. He claimed that the visual process happened accidentally. We know more about the visual process than about any of the other sensorial processes, and so I'm really pleased that we have Lauren Sumner-Rooney of the Museum für Naturkunde, because she will tell us a little more about these processes. Our third topic is devoted to transformation. Transformation from analytics of the sensoric processes to application. The application of sensorics to different fields. It was mentioned already that optogenetics comes in here. The sensorics we know from plants, animals, and the like are used to study different processes, for example, in neurosciences. So I'm really happy to have Hannah Monyer here. She has given excellent lectures on different other occasions. I was always thrilled by her overwhelming enthusiasm, and I welcome her most warmly. I'm looking forward to her presentation. So this takes me back to where I started. Mr. Häusser, you need to take over now, and I'm looking forward, together with you, to the presentations.
Ganz herzlichen Dank.
Thank you very much, Professor Hegemann. Thank you. And we also have to thank you for organizing this symposium. You are our host tonight, to be precise. So we heard a lot about photosynthesis already. We heard a number of terms floating about already, and I must say that I studied photosynthesis some time ago. We did a video and we tried to produce such a cell. We used a tea bag and toothpaste and that sort of thing. And it was quite interesting that we created a kind of voltage, and we thought, well, we understood something. No, we didn't understand anything, which I noticed when viewers send us questions, and they told us that they repeated the experiment, and what happened then, and can you explain me? And I thought, well, I can't. I hope that I will understand more now, because when I heard in the preparatory talks that the process of photosynthesis, which is often mentioned also in your daily life in school and wherever, that this process has not been understood for a very, very long time. And not so long ago, the first bits and pieces were decoded. So obviously the video has fulfilled its purpose. People look at the topic more closely, and now we can all learn a little more. But it's an essential process. So much we probably can agree on. The world is a blue planet. Perhaps we can look at that together. Oxygen that makes life on Earth possible largely comes from photosynthesis. This is how it was produced. And we should talk about what else we can learn from the process. Once we know it really well, perhaps we can use it for different purposes. Perhaps we can do something like, well, producing green hydrogen, and maybe help to solve the big climate problem we are all talking about now. So a lot of opportunities exist in that connection. So let's the Prof speak now. I'm very happy that Professor Athina Zouni will now give us more details of the process and topics. Thank you.
Yeah. A warm welcome from my side. Warm welcome to Adventure Photosynthesis, the title of my presentation. Before starting, I would like to ask the audience a question. Do you have any idea how much oxygen you consume per hour while sitting here? What do you think? How much oxygen do you use up within 1 hour? No idea? Let me tell you, 25 liters an hour. And if you would move, you'd use up even more. And it is even more fascinating to find out that this fundamental water splitting mechanism that Professor Hegemann has already mentioned, where oxygen is released in the process of oxygenic photosynthesis, has not been 100% understood until this very day. That is quite interesting. The question is, why is that? We will now take a deeper dive into bear with me. We will take a deeper dive into the process of photosynthesis. Of course, we will start with the evolution. Around 2.6 billion years ago, cyanobacteria have evolved. That means they were already able to conduct oxygenic photosynthesis, releasing oxygen into the atmosphere. And as we know from our days in school, that is the basic underlying principle. A simple reaction. Cyanobacteria take up water molecules they take carbon dioxide from the air, and by using sunlight, they form carbohydrates, and they release oxygen through the water splitting reaction. And later, over the course of the evolution, more organisms have evolved. All of them were able to conduct this oxygenic photosynthesis and thereby releasing oxygen. These organisms have contributed to increasing the content of oxygen over a course of 500 million years, and the content of oxygen in our air has remained at the same level, around 25% in the atmosphere. The organism shown on this slide, they have a biochemical system that is able to oxidize water, and we will now have a closer look at it on the next slide. What we see here is thermophile cyanobacterium, which we've seen before, and that is what we work with in our labs. They can produce oxygen, and we will take a closer look at one of those cyanobacteria to have a look at them and what's inside. What we see are phycobilisums, external antenna systems that take in sunlight, solar energy, and transmit it to a thylakoid system, which you can see here, to a thylakoid membrane where 4 membrane proteins can be found or are embedded. The first of all is the photosystem 2, where the actual water splitting reaction happens. Then we have cytochrome B6F and ATP synthase. In the light reaction, because that is what happens here, what we see is that all 4 membrane proteins are part of this process through a linear electron transfer and a proton gradient that moves along or that develops through the membrane, and therefore the ATP synthase, highlighted in red at the right-hand side, ATP is produced, high energy product. So these are the basic principles underlying this reaction, and we will focus on the photosystem 2, PS2, where this fundamental water splitting reaction takes place. What we see here on the left-hand side is that with light, photons in this case here, we see that 2 water molecules are split into 4 hydrogen ions. We see that oxygen is being released, the oxygen we breathe, and one after the other, the electrons will move to a mobile acceptor. That means they're being taken up through QB, the plastokinin, and it is then being reduced. And in the following, we will have a closer look at this reaction, because that is what we're interested in finding out more about. The adventure photosystem 2 started in 1995, at least for myself, so quite some time ago already. The aim of my project at the time was to generate photosystem 2 crystals that were oxygen active at the same time. In 1995, there were many doubts about whether this would be possible at all, because there were only a few membrane structures we knew of at that time. So that was the major challenge we were facing. It was a risk, of course, and what other scientists said to me at the time were that will not work, it doesn't work, it's a waste of time, etc. So that was what my fellow scientists and colleagues told me. Personally, this was a challenge, because I found that this was quite an exciting field of research. There weren't many structures of membrane proteins known at the time, which I found exciting, and that is why I wanted to find out more about it. And I will try to be brief and concise, because the process is quite complex until the actual crystallization. And what I was able to achieve is I was able to generate these oxygen active PS2 crystals with a size of 400 micrometers, and using light here flashes, you can use an electrode, an oxygen selective electrode, and you can even prove that there is oxygen here. And that was only the first step for the next steps. The next step was to define the structure, which again took a few years, because we had to improve the crystals we were working with. What this slide illustrates is the photosystem 2, a dimeric molecule. We used a resolution that was around 3.8 angstrom, And what we can see here is this fascinating molecule consisting of two monomers and 20 subunits for each monomer. And apart from that, in the luminal area, we can see that there are 3 subunits. We see chlorophyll, carotenoids, and lipids. But the most important thing, bear with me, the most important thing was the manganese cluster, where the actual water splitting process happened. This is a catalytic cluster, and the resolution wasn't great, but we were able to see the metal ions, and we already knew that we were seeing manganese here. And of course, that was a great achievement, which we celebrated because that was a highlight in photosynthesis research, the very first structure of photosystem 2. 10 years later, colleagues, our competitors from Japan, overtook our research by finding an even better resolution, 1.9 angstrom. That was only possible by improving the quality of the crystals they were using, and apparently they succeeded. What we see here is 4 manganese molecules in violet, a calcium ion in yellow, 4 water molecules that are directly coordinated with manganese 4 and calcium, and surrounding them we see residue of amino acid, which is of course embedded in a protein matrix. This complex is called oxygen evolving complex, OEC in short. Why? Because oxygen is being released in that complex.
Ja, aber man hört mich noch, oder?
I hope you can still hear me. Great, thank you. And following that, we wanted to find out more about the dynamics of the water splitting reaction. There is a model developed by Mr. Koch called the Koch model, and he took a closer look at what actually happens within the manganese cluster, and I think we should also have a closer look at that. The OEC, the oxygen evolving complex, is seen here with 4 manganese ions, and he said that if the crystal is in a dark state, we have 2 manganese with an oxidation number +3 and 2 with +4 in a dark state. That, by the way, is the most stable state the manganese cluster can be in. And now we apply light, we expose it to light, and what we found is a P680 consisting of 2 chlorophyll, where we have a separation of the charges, and 1 electron is transmitted to the acceptor molecule that I've already mentioned. What happens now is that at PD1 and PD2, as you can see here in the middle, an electron gap is being developed, which needs to be filled in with something else. What happens now, consequently, is that a reducing active molecule, this electron is then fed into the system again. And what happens next is an electron is being split away from the manganese cluster, an oxidation reaction, or in other words, the manganese cluster is oxidized once. And second flash of light, what happens now is exactly the same procedure. A void or gap is being produced, another electron is being transmitted into the void, and another oxidation reaction happens where 1 proton is being separated from the cluster. And The QB I already mentioned, which is mobile, takes up 2 electrons from the cytoplasm. It takes up 2 protons, goes into the thylakoid membrane, and a fresh acceptor is being built into the system again. And third flash is applied. Same thing happens again, and what happens now is still unclear. That is the last oxidation step, reaching the S4 state. It's a very brief state where we're not even sure whether it actually exists. And what happens then is a spontaneous reaction without light. Two water molecules are being split, oxygen is being released, and the electrons are being fed into the manganese cluster, which is the most reduced form of the manganese cluster. The fourth flash of light is applied, and the cycle is then closed. What did we want to know, or what are we trying to find out about that? What happens between the different stages or states? What happens between S1, S4 and SO? When is oxygen produced, or how and when is it being released? That is the question we're dealing with. And is there even a state 4? And the next question is, where do the water molecules come from that are used in this reaction? Still not clear either. With the classic X-ray structure method, one crystal is being examined, and what you see here in the left upper corner is the crystal that we were using. You also see the holes that were hit by the flash, and of course the question was whether this protein, the manganese cluster, was still in state 1 or whether it had already been destroyed. What we found out is that there were radiation damages. In other words, we weren't able to continue our research because we needed the entire structures, and if the manganese cluster had already been destroyed, there would not be any sense and would not make any sense to continue. In 2011, there was a revolution in the area of crystallography because the femtosecond X-ray free electron laser was developed. XFEL in short. What is the advantage here? Femtoseconds means ultra fast, 10 to the power of 5 seconds, 10 to the power of 6 seconds, and in 40 femtoseconds this X-ray pulse was used, developing an X-ray image, and within that amount of time nothing happens within the crystal regarding the proteins and the manganese cluster. In other words, this method made it possible to measure the crystal without using X-ray and at room temperature at the same time. But there was one main disadvantage, which is we were only able to measure one crystal with one image, and then we needed more crystals, and in fact we need at least 100,000 microcrystals to measure one complete data set. My former PhD student, Mohammed Ibrahim, had a closer look at that. It was one of the goals of his doctoral thesis, and he actually has accomplished he was able to generate microcrystals in a large amount, which were uniform, especially within the core of this crystal. But he also had to develop an injector system, which is shown in the middle of the slide. We see a droplet on tape experimental setting. The setting is quite easy to understand. Have a look at this video. The droplets are applied to the tape, And here you see the reservoir where the crystals are being contained within, and the visible laser beams that can be applied here. You also see the free space laser that is being applied, and after that we use the free space laser, and on the screen we can see every single crystal, and we have an X-ray image for each of them, and these images can give us an impression of how well these crystals have been analyzed, and then we can have a further look at these images. So we've had a look at the different structures, the X-ray structures of the manganese cluster, of the different states, as 1, 2, 3, some interim values as well, about the manganese cluster, of course. And we had a closer look at the integration of a water substrate. So we had a look at S2 and S3 in order to then proceed to the more exciting phase where oxygen is being produced and released. Let's have a look at S2 and S3 and the transition from the second to the third state. Here we see 2 manganese, 1 oxygen. So obviously the reaction has not happened yet, and in the third state or prior to the third state, what we see is that between the first manganese and the second and calcium, that an oxygen molecule has been formed. We see this water molecule, but a proton has left this molecule as well, which means that we have an OH at the end, hydroxyl group. Okay, we'll give it one more try, or we just use a microphone.
Das geht jetzt, ja? Okay, gut, ja, ne? Kleine Verzögerung.
Sorry for the delay. So, what was I saying? We were wondering where these water molecules came from and where those protons were moving to. My former PhD student, Ms. Hussein, has had a closer look at this question. That was the aim of her doctoral thesis. In the middle, we see the manganese cluster, and we also see different channels. For example, O1 channel, O4 channel, these are water channels, and we see a proton channel marked in green. And of course, that made sense, because water had to be transported to the manganese cluster, and protons had to be transported into the lungs. She's had a closer look at the different channels and found that close to manganese 1, the molecules were very flexible, very mobile. In other words, it was like a mill, and we assume that one water molecule was then transported to the manganese cluster, and one water molecule is then being transported after that. She's also had a look at the proton channel and found out that there's a bottleneck. If you have a closer look at this area, you will realize that 2 amino acid residues are in this area, and over time you can see that they act as gates, opening, letting protons through, and then closing, blocking out more protons. So this is a very controlled reaction. moving on to the most interesting question. What happens between the third and fourth state, which is binding of O2 and release of O2? At 700 microseconds after S3, what we realized is that O5 and O6 still exist. They are neighbors, very close to each other, can bind to each other and can be released. When O5 leaves this system, then it is most likely being replaced by a water molecule that is very, very close in the proximity here. That is just one model. Of course, there are more models, but we believe that this is the most likely one. And at 1,200 microseconds, we realized that O6 was gone. In other words, this could only mean that O2 had been released. And we then came to the assumption, and I highlight assumption, that there was a delay between 700 and 1,200 microseconds, and that there was a very brief intermediate state in the fourth state. So these were the most recent results we've just recently published. So what are the lessons learned from the natural water oxidation? Well, what Professor Hegemann has already mentioned, we are striving to find different ways of applying our results when it comes to using solar energy, climate change, finite fossil resources. Of course, our aim is to be able to use solar energy in an even better way and using it for converting solar energy into chemical or electrical energy. Our long-term goals include artificial photosynthesis, also the production of hydrogen, for example, combined with other enzymes, or, and that is also important because we engage mostly in basic research in order to understand these reactions. And of course, nature is so complex, we cannot copy it one-on-one. What we can do is copy principles and different modules in order to apply them into our research. This is a great work accomplished by our Chinese colleagues, and what they did was they developed an artificial cluster based on the natural cluster they found in nature. In conclusion, I would like to thank Professor Holger Dobbek, who is here with us today. We've been cooperating for quite some years when it comes to SFVs and UNICAT. Johannes Messinger, professor in Sweden, and I would also like to mention a close cooperation for more than 20 years with the scientists from Berkeley. You see them here, and looking at these photos, you can see how many researchers are involved in this process of decoding these underlying processes. And last but not least, there's a philosopher I would like to quote: all new ideas pass through three stages. First, they are being ridiculed, then they are being violently opposed, and thirdly, they are accepted as self-evident. By Arthur Schopenhauer. Thank you very much.
Vielen herzlichen Dank.
Thank you very much, Professor Athina Zouni. One day we will not smile at the acoustical problems, they will be solved. Unfortunately, we have a full program, so I will be briefer than I'm usually known for. We now leave that area, we zoom out and go elsewhere, namely to things a bit bigger. Sea urchin was about that size the last time, at least when I stepped into one. So what does a sea urchin do with light? He he or she, that it has not got any eyes, at least not the kind of eyes we know. But sea urchins do see light. For example, when a shadow approaches, they run away. They orientate, they navigate their environment, although they can't see. So nature has developed a very efficient way of giving this animal an opportunity to perceive light, which is quite funny. Just imagine self-driving cars running on green hydrogen, and they have all sorts of radars and lasers and ultrasound and all sorts of advanced technology, cameras, of course, sensors and whatnot. The sea urchin doesn't need that much technology. Nature uses what is there, and how exactly it works, we will learn from Dr. Lauren Sumner-Rooney. Welcome.
Thank you. Thank you very much for the invitation to speak this evening. First, I'd like to apologize that I'm giving my presentation in English, but I think it's better for everybody than my trying to do it in German. So please bear with me. I'm reassured that we have fantastic translators working on that. So we've heard a bit this evening already about light as a source of energy. And this incredible fundamental reaction of photosynthesis, which is the basis of pretty much all life on Earth. But I want to talk a bit more now about light as a source of information. So almost all animals on Earth can sense light in some way. And for more than 500 million years, animals have been using this light to gather information about their surroundings. Now, this could be to find themselves a mate, or learn what time of day it is, when they should be sleeping, when they should be active. They can use it to detect predators or navigate through their environment. But universally, light is an extremely important sense, and light sensing is extremely ubiquitous throughout the animal kingdom. So how do we detect light? How do we turn this energy into information, into nerve impulses, as it were? So throughout our bodies, we have cells for sensing all sorts of different energies. We're going to talk about photoreceptors, so these light-sensing cells. The majority of light sensation within animals depends on a family of proteins called opsins. So these are G protein-coupled receptors, and they bind a smaller molecule called retinal, so as you can see here. When a photon or a group of photons strikes this opsin, you actually get a transformation in that retinal molecule. So it transforms from being in this cis position. You can see there's a little kink in the molecule here. And that transforms into trans-retinal, thereby triggering a change in the conformation of that opsin molecule. So you've gone from having a purely energetic photon all the way through to having a change in conformation of this protein that then triggers the response of the photoreceptor cell and further neurons. And this doesn't just happen in our eyes. I'll just be talking about eyes this evening, but it happens also in our brains and all over our bodies, particularly in other animals, all over their bodies as well. So there are animals that have light-sensitive skin, for example. We have photoreceptors within our brains. It's a very common sense. But what is vision specifically? So vision is not just sensing light, being able to tell whether it's light or dark. If you imagine one photoreceptor by itself exposed to light from all sides, it will not be able to tell you anything about your environment other than whether it is light or dark. However, if you start to add pigment to that cell, surround it, then you'll actually be able to start telling which direction the light is coming from. And gradually, if you add more cells, they can tell you, little by little, more about the spatial information that you have. So vision really must give you spatial information about how light is distributed through your environment. Now, this example only gives you 4 pixels, as it were, if you have 4 photoreceptors. If you add more, if you add lenses, if you end up with a very large population, you can start to extract more information about your environment. So the more sensors you have and the smaller and more distinct the area they sample, the higher your resolution. This ability is actually very ancient. So more than 500 million years ago, when animal life was exploding on the sea floors of shallow seas, this is when we start to see evidence of vision in animals, and it changed everything for evolution. So you start seeing animals eating each other, moving around, exhibiting directionality. And one of the potential triggers for this is the evolution of vision and the evolution of eyes themselves. Since then, eyes have evolved more than 40 times in a huge range of different animals. So some of these will be familiar to you. Obviously, you can see our own eyes. You might recognize the octopus, crustaceans. But actually, some of these are more surprising and in more unfamiliar places. This is the area that I'm particularly interested in. Animals that have weird eyes that you wouldn't expect. Maybe the wrong number, maybe in animals without a brain, maybe distributed across the body. For me, this is the really interesting part of how animals, unlike us, see their world. So how do we measure vision? What are the important factors that could contribute to how an animal sees the world around it? There are 5 key factors here that I'll just walk you through very briefly to give an idea of exactly how we might quantify our own vision and then how we might apply that to others. So the first and possibly the simplest is what we call field of view. So where is your eye looking in the world? How many do you have, and where are they all looking collectively? So this is probably a less familiar visual system for you all. This is a chitin. They're mollusks that live on the seafloor. And you can actually see that they're covered in tiny, tiny eyes. Across the whole body, you might have about 400 or so. But each of these eyes has its own field of view, and spread across the entire animal, you have a very dense, very complex sampling of the world around them. So these guys are really the kind of champions of gathering information from every degree around them. They sample and sample and sample every bit of space for these guys to make sure that there are no predators coming by. We might also look at resolution, so how fine a detail can be resolved. Initially, we think about spatial resolution, so you might be familiar with jumping spiders. These guys have very, very sharp vision, very high acuity. So if you imagine resolution being measured in terms of degrees, the same as angles, so if you extend your arm to its full length, the width of your little fingernail is subtends about 1 visual degree. But these spiders are operating at a fraction of that. They're the highest resolution known outside of vertebrates. So high that they can not only eat other jumping spiders, but they can also look at and solve visual mazes before they get there. We also might think about temporal resolution. So how quickly can something see? You might be familiar with the frame rates that you need to make something appear like it's moving. So film depends on having 25-ish frames per second. Some animals don't need that much speed. If you're a starfish, for example, you might only need to see where rocks are nearby, and they're not moving anytime soon. So you don't need very high resolution in terms of your temporal vision. But if you're a predator, if you're intercepting flying insects or flying birds, then you have much, much faster resolution vision than we have. So both of these are multiple times faster than us. We see at about 40 hertz. But if you look at a peregrine falcon or a rubberfly, they see much, much higher resolution. Contrast in terms of how light or dark things can detect is also very, very important, particularly when you're thinking about different light environments. So the most sensitive to changes in light intensity is known from these ogre-faced spiders. So there will be a couple of spiders appearing in this talk, arachnophobes, beware. But these guys have the incredible ability to spin a web that they use as a gladiator's net to capture prey in complete darkness. We're now moving on to things that we can't always sense ourselves. So first of all, we'll talk about wavelength. So we've spoken a little bit already. We've heard from the first talk a bit about light. But if we think about how long the waves and the wavelengths of that light are, this is what we interpret as color. So we only see a relatively small portion of the spectrum of electromagnetic radiation. There are actually animals that can see beyond that either side, so into the UV and into the infrared. This also might be a familiar creature to some of you, the mantis shrimp. So these guys have extremely high numbers of different opsin molecules sensing different wavelengths. It doesn't only affect how animals see their world, but how the world then evolves to appear to them. So this is a series of images taken of flowers that you might go out and find in any Northern European area. And then they're viewed on the right-hand side through a filter to extract the UV. So this is an instance where bees and other insects being able to detect UV has influenced the evolution of the flowers themselves in order to highlight particular areas. You can see that they're drawing the eye towards the center where the nectar and the pollen are found. Finally, the most alien of these properties of light is polarization. So if you imagine these waves of light, the length of which we've decided is color, the oscillation can only occur around one plane at a time. So if you have oscillation around multiple planes, you have this kind of scattered, unpolarized light, which is what we see. If you imagine filtering that through, for example, polarizing sunglasses, then you'll only see the photons oscillating along one plane. To us, this is pretty much invisible, but to some animals, it's not only visible, but highly useful. So this is cuttlefish. These guys can not only use the reflected polarized light of their prey, so particularly shiny surfaces, silvery fish can reflect polarized light, but they can also use it to communicate between themselves. So it's hypothesized that actually some cuttlefish give off specific signals using only polarization and unpolarized light. This means that only other animals able to see this will detect those signals. It could be a perfect way of communicating secretly without somebody else eavesdropping nearby. So all of these properties of vision depend on morphology, physiology, the connectivity of the system, but also on each other. So there are trade-offs to be had here. You can't maximize everything, or if you can maximize everything, you're going to need a really big eye. It also depends on the ambient light intensity and the spectral content. So if you're in the dark, it's much harder to detect detail, for example. You'll notice that if you walk into a dark room, you can't see quite as well as you could before. This is influenced by all sorts of different aspects. So one is the eye structure. So this is a schematic showing the two broad eye designs, as it were, a single lens eye like our own, and compound eyes that we find in arthropods and many other invertebrates. After a century or so of research, we've actually worked out there are some quite consistent geometric relationships between some of these different factors, and we can use those to predict how an eye might function. So for example, if you have a large pupil, a large aperture, as you can see here on our penguin eye, you might be more able to collect more photons, have a higher sensitivity, but also distinguish higher spatial resolution. On the downside, if you pack your photoreceptors too close together, they might have to be narrow and therefore less sensitive because they catch fewer photons. There are also physiological adaptations that can affect how vision works. So we spoke about opsins at the very beginning. These can be tuned by changing the protein structure to absorb and respond to very slightly different wavelengths of light. So this is the basis of color vision, for example. Alternatively, you can have downstream processes that change how you process that information. So instead of having a single photoreceptor be activated by a single neuron, you might want to pool those signals across time. So researchers, including Anna Sterkel, who's I think a new fellow of the Society of the Academy, have worked with elephant moths to show that they actually slow down the responses of their photoreceptors the darker it gets. Alternatively, you can pool them together between neighbors. So you lose resolution, but you gain in sensitivity through something other than your eye morphology. So in terms of our own vision, what do we know about ourselves? How are we quantified? How do we measure up against other animals? So we have a relatively good field of view for both eyes. Your view is bounded by your eyebrows, your nose, and then to the side and to the bottom, it's a much larger field of view. We have a very acute fovea, so a high resolution area in the very middle of the eye. We're actually pretty color sensitive, so most humans are trichromatic. This means we could distinguish between three different sets of wavelengths. And we've got decent temporal resolution. So as I said, not quite as fast as those peregrine falcons or those robber flies, but much better than a starfish. And we're actually pretty good in the dark as well. But this obviously doesn't apply to all animals or all visual tasks. Depending on what the actual need is for vision, we can adapt how the eye works, how it collects information, and how it's processed. So if you have to interpret this very elaborate, very attractive mating dance from a potential suitor, you might want to look at the fine details, the different colors, exactly how fast he's moving. But if you're a fan worm that's just worried about a predator approaching, then really you don't need any of that. And this is what generates that huge diversity. It also depends on the light environment, as I said. So whereas you might live in a habitat that's very blue if you're underwater and only need to detect those wavelengths, you also might live somewhere that's extremely complex and bright and colorful, meaning you might need to actually sense more colors, higher detail, and again, faster temporal resolution. We can see these reflections really nicely in eye structure as well sometimes. So a classic example is looking at fiddler crabs. So you can see on the graph just to the right here, these guys actually live in very flat environments where all the important information is here on the horizon. So if you're looking out for other fiddler crabs or predators, then actually you see that they adapt that middle horizon band of their eye in order to anticipate where that information is going to come from. So if we imagine a typical scene that you might walk outside and see after this symposium is finished, we've got all sorts of colors, movements. Say, for example, you were walking down towards the Fernsehturm and you bumped into Oscar-winning actor Nicolas Cage. There are lots of important cues that you might want to pick up on here. So, for example, you might want to not get hit by the tram that's coming towards you very quickly. You might want to look at the TV tower in the background. You might want to really focus on the emotion he's conveying in the fine details of his face. And you might want to be aware of other people walking past while you ask for a selfie. But how does this look to other animals? Okay, maybe this is not a typical scene for all of these other animals to experience, but how might we imagine that they see it? Let's start with an underappreciated, in my opinion, fellow inhabitant of Berlin, the pigeon. So they might be the most similar to us on the face of this comparison, but actually there are some interesting differences between us and these guys, even though we have very strong shared roots in terms of vision. So you probably know that pigeons have a very wide field of view. They can see almost 360 degrees around them. But you might not know that they also have a very clever mechanism to improve their distinction of different colors, which is that they employ different colored droplets of oil that they place in front of their photoreceptors. So we're trichromatic, as I said. Most birds are tetrachromatic. Pigeons, I think, are functionally pentachromatic. They can distinguish 5 different sets of colors. And one of the neat tricks that they've used to do this is that they place these colored droplets in front of the photoreceptors to narrow the bands to which they respond. Something that's been used in many studies, from detecting cancers based on pigeons determining color differences, through to pigeons deciding what art movements appeal to them most. That's a real experiment. They got to choose between expressionist paintings and surrealist paintings, and they preferred the expressionists. So a pigeon might see this scene in a relatively similar way to us. Extended field of view, perhaps some more interesting colors, maybe a little bit blurrier, not so high resolution, particularly in the distance, but broadly speaking, not too dissimilar. But what if we looked at something a little bit more alien? So spiders, for example. Spiders actually have, most of them, 4 different pairs of eyes that can do several different jobs. They come in 2 flavors. So this principal everted eyes and these inverted eyes, or secondary eyes, on the right-hand side. Oops. As you can see, different families of spiders have adapted this blueprint in lots of different ways to perform lots of different jobs. But broadly speaking, they've split into 2 general groups. So the first, you might be more familiar with, those that sit and wait for their prey on a web. These 2 examples that you can see here are typical of this. You can see they have their 4 eyes arranged like a halo around the head. This gives them brilliant 360-degree coverage, and when they're very exposed, such as sitting on a web like this, it gives them better awareness of nearby predators. On the other hand, you might have a spider that doesn't hunt from a web, but goes hunting for its prey actively. This is similar to the jumping spiders and the wolf spiders that you can see above me here. And here you actually have much more concentration towards the front and much more divergence between what each eye is doing. So here we see all sorts of fascinating adaptations. Just as a couple of quick examples, we have these ground spiders, which are able to detect that polarization of light that I was talking about before. So what we can't see is that the light of the sun and the moon actually cast a band of polarization across the sky that we're unaware of. But these spiders have actually adapted a particular pair of eyes to use that to navigate back to the retreat that they live in. We've also, as we saw in the pigeons, we've got examples of jumping spiders which use filters in front of their photoreceptors to see extra colors. So while most spiders and most arthropods, in fact, can't see red, these guys have come up with a smart way of adding that to their repertoire. We also see underwater adaptations to vision. So this is the only spider that spends most of its life underwater. You can see it's got an air bubble around the abdomen here. And what we see in these guys is that they have very, very rounded lenses like the eyes of fish in order to overcome the decreased difference between the refractive index of water and the eye itself. And these are just generalizations, but within all of these eyes, we also see regionalization, as you can see here. So this is a map of every cell in one eye of one spider, colored by how large the field of view of that cell is. So here we can see, as is often attributed to things like antelope and animals living in plains, they also have this horizon of high resolution across the middle of the eye. So this jumping spider might have one very high resolution pair of eyes looking, perhaps into the center of the scene, but they're surrounded by blurrier but still very sensitive other pairs of eyes sampling around them. Now these might be very sensitive to movement in the case of a jumping spider. They're very sensitive to any small movements, and they can turn and direct that high resolution color vision pair of eyes that they need. But why are they using 4 different pairs of eyes to do this? We can do all of this with one pair. And this is actually all about size. So if you think about how restricted you are by how much light you can capture in the tiny, tiny eye of a spider, you can start to understand why they need to split these jobs between different eye pairs. The last example I'll talk through very quickly is actually something that was alluded to in the introduction, so seeing without eyes. So these beautiful animals are related to the sea urchins that we were introduced with, but these are brittle stars. So these guys are actually using individual photoreceptors that you can see here in red. So these images are generated through a process called immunohistochemistry. What we do is that we take a sample of the tissue. You can see the structure of the skeleton here. And we have antibodies that react to those opsin proteins that we were talking about at the very beginning. So where you can see red here, you can see opsin, and you can see where these photoreceptors are located. And we can see that actually we have both of the elements that we need for an eye here. We have photoreceptors, and as you can see on this image down here, we also have pigment. So this is actually an image of the same animal taken during the day at the bottom and during night at the top. So they have this very distinctive color-changing behavior that they go through. When we actually decided to test whether or not this does function as an eye, we performed a series of experiments where they tried to orient to a band of high contrast. So as I said, determining between light and dark is not enough to be called vision, to be called an eye. In these cases, we tested whether they were able to extract this spatial information, this black and white stripe, from this non-spatial gray surroundings here. And we found that actually they were completely able to do this as long as you tested them during the day. So this is only when they have this pigment surrounding their photoreceptors, as we predicted, but it does mean that we have a completely new fundament in these brittle stars and these sea urchins for what it means to have an eye. How on earth they process this information is still somewhat mysterious. In sea urchins, it's thought that being round has something very simple to offer in terms of comparing information from different directions, but this is yet to be tested in brittle stars. So although we're very excited about these new kind of eyes, the scene to a brittle star might look extremely blurry. We're seeing very low resolution from the behavioral experiments that we're running, but they may well be able to detect things that move. Plus, if you were a brittle star in the streets of Berlin, you're likely to only see the tram and Nicolas Cage as a direct threat, and that's all you need to do. So we're often asked, why do these animals have such bad vision? And what has the best vision? I think this is a very typical question of a lot of evolutionary adaptations, but I would argue that neither of these is really necessarily better. If you imagine being given these 2 cars and being asked which is better, it's really key that you have the information that you need to talk about which task you're going to fulfill. It's really important that we don't just focus on animals that have the biggest, the best, the brightest adaptations. Often it would be a waste of energy and a waste of resources to make your eye bigger or better than it needs to be. So very briefly, why should we then keep studying animal vision? I've given you a small flavor of all of the diversity that is out there, but why should we kind of continue to explore it, and how much is there still to know? Fundamentally, we cannot understand the world around us if we are limited to our own perception of what it looks like. So just two very brief examples. First of all, we can't understand how animals communicate with each other. So as I spoke about UV signals earlier, this is actually something that's not restricted to flowers, but it's also used and exploited in spiders. Polarization-based signals in cuttlefish and stomatopods can be really important to how they interact with each other. And actually, how animals navigate through their environment using polarization patterns in the sky or using potentially even magnetic fields is all reliant on vision. More selfishly, and often people ask for more applied reasons why we should continue to work on these, actually light is a really important impact that we have on other organisms as well. So light pollution is extremely widespread, it's growing, and we can't replace it with anything else. Whereas we can stop producing CO2, or we can try and generate green hydrogen, as we've heard, we can't replace light. And so we need to know how we're impacting animals around us. And then finally, obviously, as we have heard a little bit about, we can be inspired for ourselves, for our own gains. So this is the best piece of targeted advertising I've ever received. And hopefully it reflects a phenomenon that you're somewhat familiar with. It's now impossible to buy a smartphone with fewer than 5 cameras. And this really nicely illustrates my point about small visual systems, is that you need to have several differently adapted structures to do these different jobs if you're going to be so small. So there's a lot that we can learn, a lot that we can steal, frankly, from evolution in order to be able to make our lives easier. Our sensing better, our cameras better, our self-driving cars safer, and all of this is a very exciting world that we've only just really started to explore. So I hope I've demonstrated a little bit about the incredible diversity that's out there, not just in terms of what animals see, but what we need to learn about them to protect them and to help ourselves understand the world a little bit better as well. So with that, I will thank everybody for their attention and for allowing me to go slightly over time, but also to everybody else who's helped me along the way. So first and foremost, my group and my funders, but also many wonderful collaborators that I've had the opportunity to share this with. Thank you very much.
Thank you very much for this wonderful. Vielen Dank für diese fantastische.
Thank you very much for this exciting insight into your work. As I said before, we're running out of time, which is why I will try to be even more brief and concise. Quick reminder, we will have a short discussion around Q&A session, and if you've already come up with a couple of questions, try to write them down, remember them, and for those of you who are with us virtually, please ask your questions via the chat. We will try to make sure to answer some of them as well, and I'm sure that everyone would be interested in knowing about what happens within our brains while listening to these presentations. Neurons are firing along, but you know, it'll be great if we could just switch on the light and have a look into our brains. And something similar awaits us now. Professor Hannah Monyer, welcome. The floor is yours.
Ja, vielen Dank.
Thank you very much. Thank you very much, Peter, for having me. It is a great pleasure and honor to be with you here tonight to talk a little bit about our research and also to talk about developments that have been accomplished over the past decades. And Professor Hegemann was, of course, or has, of course, played a vital role here. And tonight I would also like to touch upon different areas that show how complex it can be to find different results, to have a shift of paradigm in research. And I will talk about how I have a closer look at our neurons to learn about our episode-based memory. What is that? This is what compromises the memory you have about where you were. For example, when you will think about tomorrow, that is this memory I am talking about. And I would also like to talk about how light helps the neurons in our brain to form a memory. What can you see on this slide? Something to wake you up, perhaps? A dog, correct? You can see a dog, and since the psychology of shapes, we understand how this works. We need to know what a dog looks like. We need to know about the shape of a dog to recognize a dog. And I will now tell you about how our brain does that. We've talked about vision. The visual cortex is comprised of highly specialized cells. For example, some are active when they see something round, such as an apple. We have cells that can detect the color red or yellow. And what this slide shows is that the same cell that can detect round shapes can be part of an ensemble. When it fires together with a yellow color detecting cell, then this cell is part of a representation that will then tell our brain that we're seeing a lemon. And if a cell again fires, emitting an action potential detecting the color red, it becomes part of a different representation. This slide therefore highlights that one and the same cell means something different to us depending on the ensemble it forms part of. We only have different instruments. For example, when we detect a lemon, hundreds of different activities are involved, and the cell can recognize what we see. This is called temporal coding, and our brain contains of different cells producing a rhythm. This can be detected by an EEG, and I will show you some other examples in the course of this presentation. Such ensembles are so important because the action potentials take place in a certain phase of such a cycle. It is not only important that a cell fires this action potential, but it is important to know what other cells form part of this ensemble. The structure that we work with is a different structure. We do not work with the visual cortex. I just wanted to illustrate it because it is easier to understand, but we work with the hippocampus. You see this yellow structure here. We have one of these structures in each of our brain hemispheres, and it is important for forming this episode-based memory. In my lab, we do not work with the cells that are important for the representation, but rather with smart cells that can synchronize hundreds and thousands of cells so that they're able to build or project such representations. If you listen to music, there's an orchestra consisting of different instruments, and they play well together because there's a conductor coordinating the different players and instruments, reaching a great musical piece. And that is what we do in our lab. We work with these conductor cells, an organization of many different neurons that are active in the visual cortex as well, making sure that the cells are forming part of an ensemble so that your brain can recognize a dog, an apple, or a lemon. So these conductor cells are vital for such a recognition process. They do not do as much as other cells, but are important for this temporal decoding process. This is what happens in the hippocampus. This is the Heidelberg castle, and we have here a pyramidal cell in the hippocampus that is active together with the cells highlighted in gray, forming part of this representation every time I am in this position. And the same black cell can cooperate with other gray cells to form part of a different ensemble, and in this case represents a different image, for example, the old bridge. That is what happens in our hippocampus, a spatial encoding process telling the animal and human beings where we are, and the majority of our memories of the episode-based memory take place at a certain place. And one day from now, tomorrow, you will be able to remember where you were last night here in the lecture hall, and for doing that you need the hippocampus. So this slide was meant to illustrate that the same cell can represent one thing at one moment and something entirely different a couple of moments later, depending on the ensemble it forms part of. And to make sure that these cells fire very accurately, we have the so-called interneurons. These are inhibitory cells that accomplish the synchronization process in milliseconds, making sure that these cells fire at the same time in one ensemble. How can we measure that in animals? We introduce such electrodes into the hippocampus of such animals, and we then measure the brain activity of hundreds of neurons, and we have a much better resolution here. So the mouse here is orienting itself in this box, and we then measure what happens within the hippocampus. A colleague of mine has received the Nobel Prize for this work by describing the so-called place cells that are active when the animal is at a certain place. That would also apply here to our situation. When I'm moving here on the stage, when I'm moving here, other place cells will be activated. When I'm moving back, other place cells will be activated. And by doing that, the hippocampus is drawing up a so-called map. And you see these red dots here representing action potential that are applied for the different places and are then encoded in order to represent this place. What you see here are 3 place cells representing the movement with so-called heat maps. You see where these cells are active, and by doing that, the brain is developing a map, a spatial map of the room I am moving in. You can see further examples here, and apart from that, what we see here, the animal is now moving within this space. In gray, you see the different pathways, and the red dots represent the action potentials. And the 2 images on the right represent the different potential again. However, colleagues found a different way of encoding, which you see here on this picture, a different form of spatial encoding. And what you will be able to see here is that the same cell fires at different places, and they are called grid cells. In our brain, we have, as you might say, a GPS system. The place cells tell you where you are, and the grid cells, we were able to demonstrate that they are very important for measuring the distance I've covered. While moving, my brain can calculate whether I've walked for 1 meter, 3 meters, etc. And the faster I am, the faster these cells fire, because I need to encode the distance covered, regardless of how fast I've been moving. In this formation, in the hippocampus, we have place cells, we have grid cells, and we have other examples of even more cell types, so-called border cells, because we need to fix this cognitive map somewhere, and every time we need to define the borders of these maps. So every time I reach a wall or something else limiting my map, the hippocampus fires border cells. And then there are so-called object cells for encoding the objects that I am encountering while moving around. For example, while seeing a chair, for example, an object cell will fire. And it is through the cooperation of these cells that our brain can form this map about the place, the distance, the borders, and the object. that is what happens in the hippocampus. And that is the first step in understanding our episode-based memory, understanding what happened where and when. And without the hippocampus, we would not be able to learn. This was shown by well-known scientists in the 1950s and 60s when a patient was analyzed and studied. They had their hippocampus removed and were not able to acquire more knowledge. And that was very important and insightful because it made us understand that we need the hippocampus to learn, and we need it in order to understand what we see in terms of our spatial context. Coming back to our conductor cells. This slide shows the red cells, majority of our cells, and in blue, highlighted in blue, we see the conductor cells in our brains, the GABAergic interneurons, and here on the bottom, an illustration by Cajal, one of the most important scientists of the past century, highlighting different cell types and identifying them even through their morphology. What happens in the brain of rodents, also in human beings, is that we found the same results here. What happens when we move? We see a synchronous activity, as seen here, the so-called theta activity, 5 to 10 hertz, a rhythmic activity, the rhythm that is very typical for the way we move and also the way rodents move. And for example, if the animal feeds, the rhythm changes, and without even seeing the animal, we can detect the different movements of the animal by having a look at the rhythm here. And this highly synchronous activity is only possible due to the synchronous activity within milliseconds of hundreds and thousands of neurons in our brain. And in order to do that, we need conductor cells. Moving on to light. First of all, we and other scientists analyzed these rare cells, making up 20% of the cells in our brain. They are very hard to understand, and what we did was having a look at fluorescent proteins using transgenic technologies in order to identify these cells. You see a cross-section of a brain where we've highlighted certain cells that we then wanted to analyze based on their fluorescence, and we know that we only have one cell type that will respond to that by expressing fluorescent behavior, and that really helped us identify these cells. That again is an example taken from a rat's brain, and we were able to detect these cells where they're being developed, where they're being integrated into a neuronal network. And when looking at the next image here, images taken by me in New York, and what we were able to demonstrate here again constituted a paradigm shift. It was hard to publish the results because they were developed during the embryo stage, and that was quite difficult. The cells that you see here in the migration were obtained after birth. They also had different external influences by the environment, and when you try to publish results in this area, this is very, very difficult because for centuries people used to believe that this was not possible. These cells were built in the ventricles of the animals and then moved into the cortex and into the olfactory brain of the animals. These studies would not have been possible without these cutting-edge technologies. This is another image showing that conductor cells are not only communicating through chemical synapses, but they also use gap junction channels where they're physically coupled with each other. And again, since the 1950s, we've known that neuronal cells communicate through chemical synapses, but the conductor cells demonstrate a different level of communication through these synapses, and we were only able to show that by developing transgenic animals that had the fluorescent proteins that were then marked and demonstrated the process. Here you see the natural cell in the cortex of a mouse, and you see highlighted in green these rare cells, and that is only possible if you use fluorescent proteins here. I will hurry up a little bit because we are running out of time, but I would like to talk about something we were able to demonstrate in our lab, and it also contradicted the current dogma. We were using optogenetic tools here, and the fact that these conductor cells, which we and everyone else called interneurons, well, they only control small ensembles. A conductor cell in the visual cortex only controls them in the visual brain, not in the olfactory brain. What we found by coincidence is that this was not the case. In fact, there are superconductor cells controlling smaller conductor cells, which is illustrated by this schematic chart here. You see the red conductor cells, part of a local neuronal ensemble controlling that. And what we measured was rhythmic activity with different frequencies depending on the animal's behavior. But these ensembles do not have much in common. What we found out, also thanks to optogenetic tools, was that these superconductor cells existed, enabling a communication between neuronal networks that were far away from each other. And by doing that, we're even able to synchronize neuronal networks across larger distances. How did we detect that? We injected a virus into the brain using fluorescent proteins, and what we realized was that these spanned beyond the local area in the brain. We also used these technologies here, optogenetic tools, by introducing channel rhodopsin into the brain, and these channels are usually part of one cell algae and are light activated proteins. So we no longer depended on the response to light, but we were rather able to activate them by exposing them to light, and we were then able to find out how such cells could influence the behavior of these animals. We infected one area of the brain and were then able to use patch clamp techniques to measure single cells and understand how such cells behave once they are exposed to a light impulse from quite a distance away. And by doing that, we were able to detect pacemaker cells within the brains of these animals that were controlling the cells within the brain. I already mentioned the GPS system that we have, consisting of different cells, place cells, grid cells, etc. But we have the medial septum in our brain with long cells that are far away from each other, and I control these smaller conductor cells. These cells control the rhythm across large distances. And what does that mean? We know that animals and human beings use grid cells for a thing that we call vector integration. In the dark, when I close my eyes now and I work in a square, my brain should be able to tell me how to get back to the point where I started. And that is also true for animals. What we see here is the animal moving in a dark space, and it is still able to find its way home. Why is that so important? We are able to do just that, and the slide shows these so-called vector integration process. We know that patients suffering from Alzheimer's dementia, that the first brain disorders are happening in the hippocampus. That is why they have memory disorders, and a very critical variable is the vector integration, happening even before cognitive disorders. People with a low cognitive impairment will no longer be able to find their way back to the point where they started. And we believe that these pacemaker cells are part of this process. On the left hand, you see a normal grid cell, and on the right hand side, you see a pathophysiological grid cell, for example, because they have been affected, or because the conductor cells are not able to work as they do their job. So if the pacemaker cells do not function well, they cannot perform well either. As I've shown, these functions would not be possible without the different tools and technologies we have developed, for example, by marking a rare cell population, by marking proteins and channels that are active in these channels in order to carry out different tasks. And what is also vitally important is the communication of different brain areas that are far away from each other and still accomplish a synchronous cognitive accomplishment. Would not be possible without optogenetics. That brings me to the end of my presentation. Some of the colleagues that have been involved in this project and in this research endeavor have been doing that for the past 30 years, so that shows or goes to show how long it takes for us to come up with new and vital results. And what I would also like to point out tonight is that my lab has focused on normal brains and physiological conditions, and the Lautensteger Foundation has supported us in conducting our research when it comes to pathophysiological conditions in the brain. And I would like to seize the opportunity to highlight how important such projects are. They are only possible through the financial support of different foundations. These are high-risk, high-gain projects, and regardless of whether we talk about European-funded projects funded by German institutions, we always need the support of such foundations that take the risk and support us. Thank you very much.
Vielen herzlichen Dank. Wir dürfen auch direkt schon mit dem Mikrofon am besten Platz nehmen. Vielen herzlichen Dank.
Thank you very much, and I think you should hold the microphone, because I would like to ask all the other presenters to join you here on stage. So whichever chair is left, this will be mine, so sit where you wish, so that we can start our discussion here, provided you have a question to this fantastic panel. So either you ask the question digitally or here on site. And while you still wonder how to activate the microphone, I suggest you start by raising your hand so that we can coordinate questions a little. If it's okay, I would like to start and ask Professor Monyer. Thank you for this impressive talk. It looks as if you are on a fantastic way forward to find out how the brain functions. You always know as much as you don't know. So my question aims at the following. We try to copy things in the computer and see how quickly a computer fails. So where's the magic of the natural brain? Well, this is a comparison that's impossible to make. Our brain works differently than artificial intelligence. Our brain is an analog mechanism, it's not a digital one. Plus, the brain depends a lot on feedback. Everything is linked to everything else. Well, now it's changing about the newer AI. You also have feedback loops there, but to put it very simply, our brain, when you remember something, works within seconds because it has multiple codes. What I see is not just something that I've seen, but it's linked to place, to time, to other levels. And when a piece of information is living, the associations can help me to locate the memory that I don't have at hand immediately. But the brain, well, how shall I put it? Well, our memory does not remember the things the way they happen. What we expect from a machine is a bit different. I remember something, we call it top-down encoding. We remember the things we are interested in. Our memory and the way we learn depend very much on the state in which I find myself, on the emotions I have, on my attention, my sleepiness. So what you remember tomorrow of this year depends on very many different factors, and could well be that we meet in a coffee shop, in a coffee house tomorrow, in a cafe, and when we talk, it appears that we attended two different events. So a brief question to the audience. Anyone who dares to raise their hands? Yes. So please press the button. Can you hear me? Yeah. And switch it off after you've said your piece. I have a question to Mrs. Monyer, but before I do that, I would like to thank all speakers for their excellent presentations. Mrs. Monyer, Steve Hawkins showed in a number of works and book that he has a rather courageous hypothesis, namely that the organization into grid and place cells, the organization of the hypothetic memory in the hippocampus, concerns all abstract thinking, that these cells should be found in the entire neocortex. What do you think about that? In other brain structures, you do find place cells, and I try to simplify things. The hippocampus of itself, of course, is not just responsible for spatial information. It's a theory, well, to put it simple, there are two schools. O'Keefe and his school say that the hippocampus and the related structures, that these cells practically code for space. There are others, such as Eichenbaum or others, say no. In other brain areas, there are specialized cells, and we have now a paper that was already accepted for publication that shows that there is a synthetic code process for objects and space present. So to give you a simple answer, we have highly specialized cells, but they also occur in other areas of the brain. Their main base is definitely in the hippocampal region. Thank you very much. All those who are listening online, please send your question through the comment and chat function. And if there is no more hand up, I would like to ask the next question to Professor Zouni. How do you work, if I may ask? You showed us so many different phenomena, from 3D simulated molecule complexes to microscopic images of crystals doing the most amazing things. How much is happening in the lab? And how much do you do in the simulation? Good question. A lot of what you have seen in terms of the crystals, that's done in the lab. All the analysis, that's all done in the lab. So before we do measuring trips, because the femtosecond laser and all that, this is done in other labs in Stanford, for example, where there's special equipment, but we supply the material and the crystals. The microscopes are there. So what happens in our heads? Well, how you can improve things, and then you need a lot of creativity. We do not want to stop where we are, we want to get better. What I also showed you is that there are many different groups. One do an injector system, this droplets on tape thing. There are improvements there. So what we see all the time are improvements that do not necessarily come from us, but that are coordinated with other groups so that we can further develop together. Then there are theoreticians, of course, who have a very close look at the models. So we are part of a wide network, a very wide network. Professor Hegemann has a question. What you showed us today is the result of many years of work to solve the question what happens after different light pulses in different oxidation steps of the manganese cluster. Well, according to the publications that came out this year, a layperson would think, well, they're finished. The problem of water splitting has been resolved, so what's now left? Quite a lot, actually. You saw the two images taken at different times. What we cannot yet see is the oxygen that is released. We want to see that in the structure. At the moment we can't, which means that we need to generate qualitatively better crystals at 1.7 or angstrom or whatever, good ones. I don't know where the limit is. We should have those in order to be able to see the releasing of the oxygen and where it happens. This is still open. Other questions are equally open. The water molecules, for example, we do not yet know very exactly which of the water molecules play a role. I showed you models. So we are far from having finished. What is missing is still a bit. Good news, because I can imagine that many people are now motivated to get on board and do the research together with you. I think people will contact you after that. So if you watch now and have the feeling, do that. So anyone with a hand up here? Yes, please.
Dr. Sumner-Rooney, you showed these all different ways that animals can see and the different kind of eyes that they have. Now, for humans, it's known that you have a variation of eyesight. So some people can see really well, others not, and it's extremely complex what can go wrong with your eye. Do you have the same complexity in all kinds of animals, or do you study that? Is it possible to tell, like, this spider should really wear glasses, and what would you do to help it?
Yeah, absolutely. So there's certainly individual variation, and it's very important to make sure that you get replication for that exact reason. I mean, I think one of the important parts of studying animals as opposed to humans is that actually we can't get as much feedback from them as we can to a human participant. So there's a lot of noise in interpreting how they behave. So the level of variation there probably is between individuals, and how we distinguish that from the level of variation that is due to noise in behaviors that we see is quite difficult to distinguish, which is one of the reasons replication is important. But we will definitely see variation between individuals for sure, and over their lifetimes, of course. So, you know, some spiders' eyes might be very good to begin with, but we know that they do degrade as they get older as well.
Also eine Herausforderung, wenn ich das ganz kurz...
Quite a challenge, in other words. So the moderator briefly summarizes the answer of Mrs. Sumner in German. What one else should say is that when we test the animals for their episodic memory, they are of the same group, they are of the same tribe, they are siblings, they are very similar genetically, but you see enormous differences. Some learn immediately and others are simply stupid. Just as with humans. People are different, animals are different. The genetical setup is different and levels are different. It's highly interesting to see that even when we talk about very simple animals, that the variability is enormous and you need relatively large colonies, 15 per group when you speak about mice at least. So we have time for one question. There's a microphone on up there. I have a question. Well, light plays a great role in biology, but the molecules that perceive the light are relatively limited. For example, we saw many different eyes all working with retinol. Was nature not a bit simplistic, or is retinol so great that it's better than anything else? The same with photosynthesis with regard to chlorophyll. You could also consider finding other pigments. There's enough chemical variability available in nature.
First, to talk about retinal in particular, even though these eyes have evolved many separate times, it's likely that the actual chemical reaction of sensing light is fairly conserved. So this is something that I'm sure my colleagues can comment on as well. So there's probably fewer or single origins for those chemical reactions at their heart. The other part is there are other ways of sensing light. So I didn't talk about cryptochrome, for example, another example of a molecule that can be used to detect light, both in us and in other animals as well. But I think, firstly, there is this much more ancient chemical for light sensing that has led to diversification, certainly. But also, if something is successful, then evolution will pick it up and run with it, and it will rapidly become successful. I'm sure you have thoughts as well.
Es wäre vielleicht zu sagen, also auch.
Well, what I would like to add from my area is two brief comments. A neural ensemble works relatively similar with regard to the visual cortex and the auditory cortex. So in the process of evolution, you have something like a canonic circuit. Cells are set up differently, but you see in the acoustic cortex, you can see in the optic cortex, and you hear in the auditive cortex. When you look at an AI, it's interesting to see how it solves tasks and how we do. And an AI works with enormous data sets to learn how to solve a certain task. Now there are algorithms in place where you can see how does an AI resolve a task, and very often in a very different way, which is interesting. Sometimes completely senseless and pointless how an AI resolves a task, but even from that you can learn something. So what I would like to say is that solutions in evolution do exist in great number. We have lots of different modules in our brain, but how a single module works, well, certain principles are similar, and you could resolve the whole thing differently on principle. And I think when we look at an AI, we will get lots of interesting answers, given the enormous reserves that still exist. But you're right, it's an exciting topic. Dear panel, thank you very much for your talks. Thank you very much for your answers now. Unfortunately, we have to wind up now. And the honor of summarizing the evening goes to the Managing Director of Hector Fellow Academy, Dr. Judith Elsner.
Sehr geehrte Damen und Herren, es ist mir ein großes Vergnügen.
Ladies and gentlemen, it is my pleasure to wrap up this symposium. Well, at least to find a few final words, because summing everything up is simply not in my capacity. At the Hector Fellow Academy, we always strive to address major topics in science and academia, and as you've seen, We've heard about basic research, but also about different applications. And we also realize highly professional scientific studies, and that is something we see every single day, and we strive to always accomplish that. This is so important for us as a foundation, and also to promote a dialogue with a wider audience. And the fact that so many of you have shown up here today shows that we can do that. Dear audience, thank you so much for being with us tonight. And of course, I would also like to thank our wonderful speakers, our host, Peter Hegemann, for the scientific organization, Dr. Häusser, thank you very much for guiding us through the symposium, although we were running out of time, and I'm sure you would have wanted to say even more. Mr. and Mrs. Hector, dear directors of the board of the foundation, without you this would not have been possible, so thank you very much for making it possible to organize such symposia every year. I would like to warmly invite you to the next symposium. We will be in Hamburg in order to learn more about how children and adults learn, and in fact that the way they learn is different. And I do look forward to the next symposium on July 11th, 2024 in Hamburg. And I would like to warmly welcome you to join us in Hamburg or to be with us virtually online. I hope you enjoy the rest of this evening. Have a drink, have a snack, talk to someone, chat with someone. Always question what happens around you. See you again next year. Thank you very much.